We have four speakers, starting with Alyssa Barra, who is going to talk about cross-sectional versus longitudinal data for modeling waning. And I think I'll just let her get started.
Okay, well, my name is Alyssa. Thanks, Roy. I'm a postdoc at the Johns Hopkins School of Public Health. And today I'm going to talk to you about some work we're doing to estimate longitudinal tetanus antibody waning. This is not working. That's okay. Am I doing it wrong? Oh, okay, cool. Sweet. So across VPD antigens or vaccine preventable diseases, I'm going to say VPDs because that's easier. It's known and expected, that immune responses are going to wane over time, specifically antibody responses, which are often a correlate of protection for most VPDs. However, the magnitude and decay of this waning, while hypothesized, has been largely unquantified. And particularly, the most proposed mechanisms most often heavily influence specifically low and middle income countries. So a recent example here is a paper that was published in Nature Microbiology that examined waning measles antibody titers from longitudinal cohorts of paired mothers and children from China. And they observed these declining titers as expected, and they looked at factors such as maternal antibody concentration to predict titer levels by age, but they didn't look at any other additional factors that may influence this rate of decay, like nutritional status or any kind of other immune function. Next slide, please. Sorry, this thing is not working. Okay. I'll keep talking. So tetanus specifically is currently recommended in a 3-dose schedule before age 6 months. Okay, thank you. So tetanus antibodies are supposed to and are expected to wane quite substantially. And because of that, the WHO recommends 3 booster doses to ensure protection and contribute to reduced outbreaks and achieving maternal and neonatal tetanus elimination. Vaccine programs in each country set their own schedules for both primary and booster tetanus vaccines, which can often vary non-trivially between different regions. So most low middle income countries use a 6, 10, 14-week schedule for a primary dose of tetanus, but most have very different schedules for booster doses. More so than the specific timing of the booster doses, countries also have very different numbers of booster doses in their schedules as well. So, and this varies substantially by region. So, for example, in the Americas, over 60% of countries have 3 booster doses in their schedule, and no countries without any boosters. But in the African region, a similar proportion of countries have no booster doses at all, which is a lot. Okay, so vaccine efficacy and immunogenicity are known broadly across vaccine antigens to vary by specific host factors like HIV or nutritional status. And these factors, which could include malaria, are thought to impact the immune response in a handful of different ways, which can include persistent immune activation. That's not the slide. Ooh, sorry. Along with potentially skewed cellular adaptive immune responses. So a review has been published that's outlined mixed evidence of the effect of malaria exposure at the time of vaccination on tetanus immunogenicity, but shows that there can be a decreased vaccine response if the vaccinated person has malaria at time of immunization. But more broadly understudied, though, is this overall impact that malaria exposure may have on this longitudinal decay or waning of titers. So this is a biologically plausible mechanism given the wide-scale inflammation following malaria, and specifically an effect related to a lasting altered pool of memory B cells or atypical B cells following an incident malaria exposure. Okay, So to start to answer questions related to this, we partnered with investigators from the PRISM study, which is part of the East Africa Malaria Isomer. So PRISM has enrolled 3 household-based cohorts in Uganda, the first from 2011 to 2016. Across multiple study sites. So specifically here, we'll be using samples from Tororo district, which is rural and has high malaria transmission. High in this setting means people are exposed to malaria tens to hundreds of times per year. So from over 100 households enrolled in the study, all children and at least 1 adult primary caregiver were included. All participants were given an insecticide-treated net and directed to specific study clinics for all of their healthcare needs. They were routinely followed every 1 to 3 months for routine visits in the clinic, where they were screened for malaria with blood smears and other specimens collected for lab testing. And they were also encouraged to visit the study clinic for all other healthcare-related needs, where they were also screened for malaria. There's a lot of data that was collected with this study. This is just a brief touch of that, but a lot of data collected. So, like I said, blood was collected during all routine visits and at sick visits if participants presented with a fever. So samples were tested for malaria on microscopy, qPCR, DNA amplification, and other clinical features too. Samples were then biobanked to be used for other research, which to date has mostly been related to malaria-specific questions. A random subset of these samples were tested on different malaria multiplex serology panels, where tetanus IgG was very conveniently for us used as a positive control. So we have this very coincidental data set that allows us to look at tetanus IgG responses. So because this experiment wasn't specifically designed to measure tetanus antibody, no tetanus standards were included on the plates. And so from a methodologic perspective, we were very interested in whether we'd be able to convert those MFIs from the Luminex machine into international standard units. I don't have time to talk about that today. We figured out a way to do it. Happy to later. Okay, so using this very serendipitous data set that we have, our main objectives were to first explore longitudinal tetanus antibody decay, and then second, to compare the effect of malaria exposure on this decay. Okay, so here's the data that we have, and there's a couple of key features that I want to point out to all of you. So first here, we have age on the x-axis and antibody response in IU per mil on the y-axis. On the left-hand side, we have our data from children, and on the right, adults. On the top, males, and on the bottom, females. The first thing that I want to show you is this 0.1 IUs per mil, which is what we are using as our seroprotective threshold as defined by the WHO. And we can see, generally children younger children have responses above that threshold and older children do not, right? And then also adult males have pretty much just a few of them included in this study, but very, very low responses, and adult women have higher tetanus responses. So in Uganda, there is no tetanus booster vaccine currently implemented routinely into the EPI schedule. And the only other time that a booster would have been offered to somebody is women of childbearing age, right? So that's how our female responses are much higher than in the males. But for the remainder of these analyses, I'm going to forget about the adults and just look at the children. So we have over 300 kids and over 1,400 time points that we included in the rest of our analysis. Okay, so we can first take an ecological approach by fitting a distribution to each single year age group of titers. And then using the mean from each of those distributions, we could estimate antibody decay using a power model over age. And generally, we're estimating that age at seroreversion is about 4.5 years in the absence of a booster. And then we can also fit similar power models now using a random effect per individual with our full longitudinal data set. We see similar decay patterns and slope with seroreversion also around 4.5 years. Right. So as an aside, we don't always see this clear story of waning with tetanus, as we do with tetanus, sorry. which may complicate the ability to leverage ecological data to assess antibody decay. So this is another project we're working on in Zambia, which is a paired serosurvey tested on ELISAs for both tetanus and diphtheria, IgG. So tetanus and diphtheria correlation over age, which you can see on the right, slowly declines as age increases. And by age 5 is almost non-existent. Which, initially is surprising to us. But when we think about this a little bit further, it might make sense. So tetanus IgG positivity is exclusively the result of immunization. Natural infection does not provide long-lasting tetanus IgG. But this is not the case with diphtheria, right? So this work is still in progress, but we've been exploring the level of asymptomatic carriage or boosting or transmission that we might need in order to observe this age-related pattern. But really overall to say, ecological approaches limit the interpretability of responses when there may be other means of exposure for specific antigens. Okay, so related to that, exposure history complicating the VPD response of interest, there may also be multiple or frequent malaria exposures, especially in settings with high transmission, that could severely complicate our ability to use an ecological approach to ask questions about malaria exposure as well. So among children tested with samples for tetanus, which here are each a single horizontal line, there are many, many malaria infections identified. So each red filled in dot has been identified as a single febrile malaria episode, and each green filled in dot is any visit that had identified positive parasitemia. You'll notice a big transition in January of 2015, an IRS campaign was deployed within the district and basically largely stopped febrile events and generally reduced overall parasitemia in the cohort. But based on the frequency of malaria exposure and infection, ecological analyses of tetanus would not meaningfully allow the inclusion of these dynamic malaria responses over time. Okay. That's all I'm going to say about ecological data. So now using this full longitudinal set we have to first assess the effect of malaria exposure on waning, we compared decay to samples collected prior to this IRS transition to samples collected following the IRS campaign, and we didn't find significantly different decay rates. Similarly, we then compared decay among different groups of malaria exposure. So due to the complex nature of our study design and both the combination of active and passive surveillance and different treatment approaches in the cohort, we generally define malaria as the proportion of months with any observed positive parasitemia. And we also did not find a significantly different decay rate among groups of malaria exposure, across groups of malaria exposure, sorry. Okay. We also considered then only people who had not seroreverted at the time of their first sample, which was 76 children, and assessed their hazard of seroreversion by age given their malaria exposure. So on the left here, we have age on the x-axis and the probability of not seroreverting on the y-axis. And so you can see as age increases and also the probability of not seroreverting increases, the mean months of malaria also increase. And then on the right-hand side, this is the same data, just plotted in a different way with age on the x-axis and mean months with malaria on the y-axis. So when considering months with any malaria or any positive parasitemia, we did not find a significantly higher risk of seroreversion. But when we consider the proportion of months with febrile malaria only, not asymptomatic parasitemia, we do see a significant association, where children with more months of malaria exposure have higher risk of or hazard of seroreversion. And so the difference between these associations, significance might have biological explanations. The inflammatory responses associated with a febrile versus asymptomatic malaria episode are not very well characterized, but our collaborators working on the PRISM study are actively investigating this area. But it's an interesting finding that might not just be a fluke. Okay, so sorry, I didn't show you the significant one. There it is. Okay, so moving forward, we're thinking about how we can expand on this very coincidental data set that we have and design a study that might answer these questions more explicitly. So in the third round of the PRISM cohort, vaccine history was collected via cards and recall. And so most children in the cohort have been vaccinated and most have recorded card dates as well. So we're able to assess time of vaccination. And our hope is that we'll be able to test additional samples from this third cohort with known vaccination history and status to be able to continue to ask questions related to these long-term effects of malaria exposure on decay across CPD antigens, and explore other associations by vaccine type or other clinical malaria features. We're also interested in expanding on this framework to look at short-term kinetics and temporary perturbations following single incident malaria episodes. And yeah, but in the meantime, before we're able to do any of that, we're working on an extensive review of the literature on the effects of malaria exposure at both time of vaccination and long-term kinetics in terms of both antibodies and T-cell responses across all EPI vaccines. And with that, if I have any time, I'll take any questions, but wanted to thank our collaborators and specifically those involved with the PRISM cohort at the Malaria Isomer Center.
Oh yeah, thank you for talking. They're very interesting data. Do you have plans to look at, instead of malaria as the sort of exposure, look at measles as the exposure when you do that? And I ask because measles has the strongest prior literature on likely to do something across the immune system. And so having some sense of malaria, like first, could you find an effect with measles? And second, being able to put malaria in the context of measles maybe as a positive control. Is that something you've thought about or is coming? Or how do you think about that question?
Yeah, it's a great one. In the second version of the PRISM cohort, there were suspected measles cases, and we're actively working on trying to get them IgM confirmed. But I think it's a great question. It's really interesting, but it's hard to get a data set that maybe you'd be able to ask those similar questions with, but I think it would definitely be of interest. And yeah, definitely a lot of background that would support that hypothesis. Awesome. Thank you.